are moving. And so, okay, let's get started because we have less time now. So I'll just share my screen and get going, inshallah. Okay, so welcome to this Chimera and ChimeraX workshop, which is today on 27th of March 2022. And of course, I am Haaris Safdari, and also the co-host of this meeting is Sabahuddin Bhai, who's a postdoctoral researcher. And of course, let's start with our introduction. So I am Haaris Safdari, and I am a graduate student at University of Hamburg in the chemistry department, and I'm pursuing my PhD. And the next speaker is, of course, Sabahuddin Bhai. Sabah Bhai, can you please introduce yourself?
Thank you, Haaris Bhai, for your kind introduction. And as you can see on the screen, my name is Sabahuddin Ahmed, and I think many of you already know me from before. So I am a postdoctoral scientist here in Germany at Forschungszentrum Jülich, and I work with molecular dynamic simulations and protein modeling and drug discovery and work on several drug discovery projects. So, and that was kind of a very brief introduction from my side. Thank you.
Yeah. Thank you, Sabah Bhai. And before we start our program, according to a tradition of AMU, we start with the recitation of Holy Quran. So I request Sabahuddin Bhai to kindly recite a few verses of Holy Quran.
Auzu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Alhamdulillahi rabbil alameen. Ar-Rahmanir Rahim. Maliki yawmiddin. Iyyaka na'budu wa iyyaka nasta'in. Ihdinas siratal mustaqim. Siratal lazina an'amta alayhim. Ghayril maghdubi alayhim waladdallin.
Thank you, Sabah Bhai. So we start our session now, and so basically I'll briefly start with showing you the Chimera and the ChimeraX softwares, which are both of them are basically used for visualizing the macromolecules and the PDBs, for example, right? The models that people are depositing in the PDB, and of course the EM map that you are getting. Also the crystallographic model. So we'll start with fitting an atomic model in cryo-EM map with ChimeraX. Then we will have we'll discuss about AlphaFold. I think most of you now might be aware of how AlphaFold is revolutionizing the structural biology, where you just put the sequence and it's predicting the tertiary structure of a protein, and also to compare and fit it with the cryo-EM map. Then we will take a brief break, hopefully with five minutes or something. And then we will continue with the other topics that I will hopefully cover today. So yeah, I mean, also the participants are requested to please ask any question and unmute yourself and ask any question if you have, because that's more like interactive session, so that I feel like I'm in a workshop rather than a lecture, right? So that it's easy for people to ask question if you have any doubt. If you are hesitant to ask question, you can also type in the question panel, right, in the chat box, and hopefully me or Sabah Bhai will inshallah try to answer that. So let's get going. I will just open the Chimera interface just to show what Chimera looks like. So this is UCSF Chimera, and I told you that this is the previous version of Chimera. So this is the normal Chimera, and it's not the ChimeraX that I'm going to show you now. I'll be more focused on ChimeraX. But before that, I just thought to kind of show you the interface, how this Chimera looks like. So this is the Chimera, how it looks like. And as you can see, you have several options here. So this is the maps which have been previously opened. So these are the recent files. This is the place in which your map and model will open. You have command lines here. You have active model button here. And of course, you have several menus here. So I will open a few of the maps to show how it looks like. So this is, I have some of my projects in my lab, which I am working on. One of them is, of course, this ribosome structure. So this is an MRC file. As you can see, MRC is a header file for the maps. So this is the map file. So this is the ribosome structure that I'm working on, and you can see this is the volume viewer that opens along, which shows you the name of the file, of course, and this is the box, which is 400 cubic. So it's a cube and of 400 imaginary, inside which your molecule is placed. And this is the step. So this is how many voxel you take into account. So generally, when we are seeing maps like this, we of course prefer to use one, which is step one, so that you can get the highest resolution. And you can, with your mouse, and when you press left, I will come to the ChimeraX, then I will try to follow up on and use, but for now just see. So you don't need to follow this with me, you just need to see this with me. So, and this is the density map of the map, which shows you the level. So the range of the map, if you go towards the left side, on the left click and go towards this, you see this is the range of minus, the level of the map currently is minus 0.00135, and so it starts from minus 0.01 to 0.05. And so this is generally the right side of this highest density. We see the map. So you see this is the map of tri-EM, and you see the map is very noisy here, right, at this threshold. So we try to lower the threshold, and where we kind of don't see any noise. So this is the, I would say, density to. So when you download any map from EMDB, they also tell you the threshold at which you should look at this map. For example, for my case, I would say that 0.0067 is the good density to look at the map, because there's no noise as well, and you can see the features. So yeah, so this is a 50S subunit. So as you know, the prokaryotic ribosome are made up of 50S and 30S, and together it's 70S, right? So this is a 50S. So this is a large subunit of a ribosome of a bacteria, E. coli, right? So now what I will try to do is to display this. You can also change that display style. For example, in my case, I can just make it a mesh rather than a surface. And there's the map view. And now you can also open the PDB, for example. So I just fitted a 50S PDB. This is a PDB file. So PDB file is like a text file, I have to say. And but it defines the coordinates of an atom, where all the atoms are. So now you see the density is nicely fitted here in the Chimera, right? And you see an extra density here, right? So this is the part, this is a factor for which you don't see any PDB, right? There's no, of course, there is this density also, but this is a stock. This is an L7 stock protein of ribosome. So we know that this is very flexible, so people don't do any modeling here. That's why there is no density for the PDB here. But this is the factor here, which is a small factor. And you can open this. This is an alpha-fold determined structure of the molecule, and place it there, and you see it nicely fits. Of course, there's an extra helix which is coming out of the density, but this is because this loop is pretty flexible, and that's why that the EM map doesn't show any density here. But if you try to increase the threshold a little bit, you will see that, for example, you see a loop coming from that region, right? So for example, here you see this is the factor, this 3 helix, 1, 2, 3, and so it fits nicely. So one of the functions of Chimera is for visualizing, for example, this map. You can fit the densities and try to decipher the unknown density. This is one of the functions of Chimera. But Chimera, I have to say, the problem with the Chimera is that the shading pattern and the coloring pattern that has been introduced in ChimeraX is much, much better than Chimera, which I will show in a few moments from now. So what I do is I just close this and I open my ChimeraX. I'll tell you guys when to follow me, hopefully. So I open the same map, which I showed you guys in the Chimera software. So it's the same ribosome structure. I check to step 1, and you can see now, first of all, you can appreciate that the ribosome looks a little more I would say shadowy. So the image element and is more pronounced and more better here, the graphics. And also the fact that this is the ChimeraX, and now you see this is the model panel where whatever the model that you will open, model or map will come here. This is again the volume viewer, which was basically separate in ChimeraX. And this is a log where you kind of see all of the things for now, I'll just close this logs by clicking here. So this is the ChimeraX. So, and now there are many options, as you can see here. For example, molecule display, nucleotides, graphics, maps, medical image, markers, and right mouse. I'll briefly touch upon all of these things when I come to those factors. But for now, as you can see, this is the EM map. And I can also change it to soft, for example, the view, right? So the view will change to, can also change to full lightning. Now, full lightning, you can see that there's a shadow, right, on my ribosome. And that's the beauty of this ChimeraX, that you get a more like three-dimensional view in the ChimeraX. And the developers of this softwares like Tristan Troll have like really worked hard upon getting these things working. So I'll open the same thing, which is here, open, and I go to my folder where I've stored this PDB, right? I just open this PDB, and you can see this is the PDB that opens, right? So, okay, it doesn't open somehow. Okay, yeah, it opens. So it opens 2 times, in fact. So I'll just select one of those, close it. Okay, so now you have only one PDB and one MRC open. Of course, you can see that atom view. This is all atom view. So I'll just try to hide the atom view and rather show a cartoon, right? So this is the cartoon view, which I already discussed with you guys. And now, of course, you want to see the density fit, and which there are 2 options. I prefer to do one thing. There is an option of coloring change here in the map. So you click on the left click on this, and then of course you can make it more opaque, right? You can make it more transparent. And now this will enable you to understand the transparency level, and you can see how the molecule is, the PDB is being fitted into the map. So so this will allow you to do that. And that's the advantage of using this option. So you can also do one thing. You can, for example, in graphics, you have several softwares like Silhouettes, right? So it gives you a more cartoonish view of the molecule. You can even create shadows. You can go to flat surfaces like this and then see the molecule and density. For example, you see there's an empty density here. So I will open my the factor, which is aligned here already. So you see the factor that comes here, right? This in blue color, and these 3 alpha helix beta sheets kind of anti-parallel fits properly, and then there's an alpha helix running all together. So you can see it's a very fancy software. You can this is a very simple view which was there in Chimera, but you have another soft view, for example, like this, where you can visualize the molecule in more detail and as well as more clarity. And yeah, so that's the one difference that the developers have brought into this. I will also come into the movies and how you can create movies with your folders and molecules, but that is later stage. So this is the one thing to do, and this is called fitting. Basically, you take a PDB and you fit into the map. This is because in microscopy you generally get any map, and in the map you fit your PDB and try to decipher the extra density that you see along. So this is the main function that people are mostly using Chimera for. Of course, there are many other functions which I'm going to discuss. So yeah, let's start the things. So I'll close all of them now. And now I'll request you guys to please open your ChimeraX if you have installed, and that would help me to follow the things with you guys. And for that, I would bring back my presentation also in side by side, so that, I mean, it's not necessary, but the more important thing is that you see the screen on the Chimera. So Are you guys following along? Can somebody unmute and tell that have you opened ChimeraX?
Yes, sir.
So
Yes, sir,
yeah.
we have.
Okay, so it looks like this, right? Yeah, does it look like this for you guys, ChimeraX? Anyone? Can you hear me?
Yes, sir.
Okay.
Yes, sir,
So,
we can hear you. And
okay. So yeah, I mean, somebody can speak up on behalf. I mean, the problem is that if nobody is speaking, then it feels like nobody is able to follow me. So anyway, so these are the recent atoms. So this will probably, if you have not opened ChimeraX before, this will be empty. So nothing to worry about. So let's start with fitting an atomic model in the cryo-EM map. Okay. So of course, there are two things. You might know that there is an EMDB, right? So it's a database for electron microscopy where all of the maps are deposited, right? So if you type any EMDB, for example, in my case, I will type 11997, which is the code. And it's a structure of a nanoparticle, as you can see, for a COVID-19 vaccine candidate. So it's a recent structure where people have actually displayed this nanoparticle on a trimer, and you can see it's a pretty symmetric structure with a symmetry, and this resolution, as you can see, is 3.7 angstrom. So so this is the EMDB, and of course its corresponding PDB, which is fitted there, has been also deposited, and people deposit both of them, map as well as model, in the Chimera. So I'll just close it. So this is the command line where you kind of type the commands and open. You can also use this open to open any map and model which are already saved here. But if you want to open anything directly from PDB or EMDB, you have to use the command line here. And it's, I mean, with practice, once you start practicing learning ChimeraX, I think it becomes more convenient, in fact, to use command line rather than the GUI interface like this and then opening it anyways. So what I'll do is I'll open this, the map that I was talking about, which I just showed you, open 11. So you guys please follow the same thing, 1197 space from EMDB, and you open this. So this is the map which already opens up. Did you guys also open this, and can you guys also see this in your screen now if you have opened? Yeah, I'm waiting for the answer from any of the participant. If somebody can tell that they are opening this and they can see it on their screen. I cannot hear any one of you. Did you guys open this map already?
yes, sir.
You need to unmute yourself. Okay.
Yeah, you need to unmute and then speak. Okay, perfect. So I mean, if anybody's like feels that I am going too fast, and so he can stop me and also tell that unmute and just tell that please go slow or something like this. Or if you are still stuck and you're not able to open, so that it's more, you know, interactive. Otherwise, I mean, I don't understand that people are following me or not, and for the speaker this becomes a little difficult to follow. Anyway, so this is the EMDB map, and I will correspondingly open the trimer that has been fitted in it. So it's a map, of course. It's a PDB. So for PDB, you don't have to write any from EMDB or something. You can just type 1WA3. So this is the PDB 4-digit code for that model. So once you click enter, you see this thing. So you see a trimer here, right? It's a trimer 3-chain, and it's another trimer here. So it's a 2 copy of trimer that has been deposited in the PDB. But of course, you want to fit only one of those, right? So you can see that this looks like a trimer, right? Where you can easily fit here. And it's a copy. So you have another 1 copy. And so you have, I think, 20 or something. You can count how many of them. So it's a 1 trimer, for example. Then you go another one and another one, right? So what I will do is to, because we want to delete this. So one advantage of ChimeraX is that once you hover your mouse, you just don't need to click. You just move the mouse over this PDB. You can say that it sees says that it's a chain A, and you are pointing towards lysine 78, and the atom is oxygen on which you are pointing. So I can move here, and I can see that, okay, this is the C, and I'm pointing towards phenylalanine, and this is the 99th amino acid of the chain. So this is the advantage of this. So you can move over here, and you see it's E, D, and F. So you can, of course, you want to fit only one of those, we can make a copy. So for that, what you do is to delete these chains. So for delete, it's a simple option, and then this is the recognition for the chain, because ChimeraX recognizes this slash as a chain ID. And since only one PDB is open, you don't have to specify which PDB it is. It's just one PDB. So you just say D, comma E, and comma F, and you press enter, and you see that the DEF chain is just vanished from here. I think I just pulled it off a little more than normal, so I'll just bring it back. Okay, so now what you see is a map and a model, and you want to fit it. So how to do that? So first of all, I generally prefer to use a cartoon structure rather than an atom. So this is an atom view, so I just hide it, and then I click on cartoon show. So the same molecule now you are seeing in a more cartoonish view, and now you want to fit this, right? So this is the function that generally people are using Chimera most probably for. And what you do in ChimeraX to fit it, there are two options. One of those is the most common is to move the molecule, because in Chimera there was software called Seger which could actually do it more easily, but in ChimeraX it's not there. But I have to say that this approach is more intuitive and more people are using it, because Seger approach takes time and everything. So what you do is to, on the keyboard, if you have a 3 mouse button, of course, if you're using 3 mouse button mouse, what you do is to use a press control, and then click on any of this chain, for example. Now you see this chain has been selected. You can see a yellow mark because I pressed my control on the keyboard, and then I pressed the left mouse button simultaneously. And so this methionine of the chain A got selected. So you don't have to select the whole model. You can select any one of those, and it has to get selected. Suppose if you want to unselect it, what you do is to go on anywhere on the background and press control again and click here. So you see it got unselected, right? So what I do was to get it back. So I press control and select it again. So you can select any of the chain, any of the residues, okay? And now what I do is to make the map more transparent. For that, there is this coloring option. You click here, you can also change the color of the map. For example, if you want to make it blue, more towards reddish, or more towards the mixture of this, right? So I'm not concerned about this, but rather I'll play around with the opacity of the map, because that will enable you to see it more clearly where the molecules split. Okay, and I'll make it more opaque. So I have to say that I like this full lightning here, because that gives a more approach. You can also try some other, whichever is comfortable for you. I mean, you can play around with the different options you have right here. And yeah, I mean, this is the simple view, but people generally, with the advent of other options in the ChimeraX, they generally prefer to use a more three-dimensional view. But for now, I'll use this full view of full shadow, full lightning mode. And now you have to move this molecule towards this, right? You have to fit in this density into this trimeric density. And for that, what you can do is that you see this right mouse here, right? This is the right mouse. So with ChimeraX, what has happened is that the right mouse function has been kept here, all of the things. So the right mouse is basically like a free working thing for you guys in ChimeraX. And what you have to do is you have to define the function of right mouse button basically by selecting one of those. So since I have already selected one of the residues of my PDB, so the ChimeraX knows that, okay, this is the molecule that I have to move. And then I click left button, the normal clicking button of move model. So now since you can see it's move model, so now what you will do is you can press the left and you can move, right? For example, if you click, so this is the right mouse button now on your mouse. So this is the 3 mouse button which I am using. Of course, if you are on a Mac and you have 2, then I don't think then I think it will be a problem. But if you are normal mouse with a right and left and the scroll bar in the middle, then of course you have to use the right. So you can move it like this, right? So it's basically a translation. But what I generally like to do is to rather not select this move, but rather rotate. And then what I do is to use, of course, now it will only rotate, right? It will not move, it will only rotate. But what you can do is to press simultaneously, because so you also want to translate it, right? So there's a command button generally on my MacBook. What I do is that I press the command button simultaneously, and then I move it, for example, or let's yeah, here, sorry, shift. So it's a shift button. So you press the shift, right, and you keep pressing it, and then you click the right mouse button, and then you move it. You see, it's a translation now. But if you remove this hand from a shift button and then only use this, then of course it will only use this rotate model, which you have already selected, because this is what you have defined. So I like to click the rotate model and then kind of move my molecule, rotate it with my left. So this for rotation, and then you also do a translation. So I do a series of rotation and translation. Okay. So, okay, so I move it and here, and you see it's still in the 3-dimensionally, not in the good position. So I'll try to bring it near one of those trimers. Okay. Visually, of course. Okay. And now if you rotate it, it looks like more or less, right? So I press the normal left button and then I just release anything. Don't press any right mouse key. And then I try, it looks more or less fitted. Now you have two options. Of course, it's visually it looks fitted. It's not fitted, but it's nearly fitted, what you can see. So ChimeraX, there is two options to do that, to finally fit it. One of them is here in tools. So you can see the tools option here. And when you go to tools, there is something called volume data. And once you go to volume data, there is an option called fit in map. And when you click here, you can see a dialog box opening, fit and map, in which it shows that, okay, you want to fit selected atoms, EMDB, and this PDB. So of course you want to fit the PDB into this map, right? So you select this PDB, which is the ID2. ID2 is here. So ID2 is your 1WA3, which is the PDB. And you want to fit it in this EMDB map. So use just say fit. And now it kind of rotates, right? And you can see that it changes its orientation and it fits. And you can also do it by not opening this fit in map, but rather just using fit in map button. So here is a shortcut for the same thing. So once you click it, it kind of fits, where it shows that it actually moves. You can see that it takes 60 steps and it shifts a little bit, rotation and average map value. It tells you everything. So what I do now is that once you see that, okay, it looks like everything has been fitted, one of the molecule, right, is fitted. So you want to fit other also, right? So what you do is to or rather I will show you one more thing first before I go there. So I just bring back the fit in map. Okay. And when you click fit in map, it shows you the average map value, of course, at which the fitting looks the best. So this is the 0.18. So this is the threshold, actually, this level of the map that I just talked about. But of course, if you want to, there's also a function called correlation, right? So correlation is basically what a map between two maps, but this is a model and this is a map. So you cannot calculate a correlation between them. So correlation is calculated between the simulated, not an original map, but a simulated map of this PDB. So from this density, I will generate a map, and this map and the map that I will generate and this original trimeric map, this region, they correlate it. So what I do is to go to options, and then I say that, okay, use map simulated from atoms. So from these atoms, I generate a, for example, 5, no, maybe 4 resolution. Okay. And then I say update. Okay. Now, so now what it happens, as you can see, there's another map that has been created. And I want to show you guys that map, right? So because once you click there, there's another map here. And you see this is the map. And if you want to hide it, what you can do is, so this is the selection tab here. So because since I've selected one of the atoms of PDB, you remember, it's showing selected. So I'll just remove this select for now. So basically I don't select any menu. And I also want to remove this whole map for now, hide it basically. And you see this is the map and the simulated map which I generated, just a single trimer, right? The simulated map from the PDB. So the PDB is already inside. So if you remove this, you can see the PDB also, right? So this is the PDB. So the correlation is actually the correlation between this map and this full map region. So it's around 0.769, so which is around 70% correlation. So yeah, that's good. So what we do is just to make things more easy, I just select this simulated map, right, from here, selection, and then I close it. I don't want to make more confusions. So anyway, so this is this. And now what you do, because now the molecules is symmetrical, as you know. So you take the advantage of the symmetrical function in the ChimeraX. So you use sim, and you have icosahedral symmetry basically here. So you say that you take the advantage of the icosahedral symmetry, which is the, and you apply this symmetry operation so that it fits automatically all of them. So you see symmetry, and you say the map, this is you want to apply for 2, ID2, because ID2 is the PDB, and you say that icosahedral, and it's called 2, 2, 2. So basically this is the icosahedral symmetry, which which you can also find. One thing of this is that you type help and type any command in ChimeraX, and it helps you to find the function of the symmetry command. So if you type, for example, help sim, it opens up the whole functions, how the symmetry can be used, right? So all of the symmetry functions and how to use this command, you can find it easily. So in my case, of course, I know that it's a symmetry function, icosahedral. So I'll just type that sim, and of course you have to specify the model for which you want to apply, and then you say 2, 2, 2, and you see. And so it applies the icosahedral symmetry, and you see this is the thing. But now you see that there are 2 chains running around, right? Because it has initially DEF, so it has 2 chains running around in the same place. You can see one blue and one pink, right? And of course, this is the 3rd chain that came along. So this is so you want to delete those extra chains. For example, you want to fit only one chain A. So what you do is you delete, okay, and then you say slash, which is the chain B, and you say chain C. So it removes the chain B and C. Now it fits only one of those. And now you see how beautifully the model has been fitted in your map. And now when you want to actually save this fitted, because this is the ultimate goal of the fitting the map in ChimeraX, because now people want to align the model with the map and then save it, so that we know in other softwares like Coot or refinement software, when people are using, they want that, okay, I want to save this finally with this model with respect to this map. And this is called aligning the map with the model, which I already did, which I showed you. This is the fitting in the map. And then, so for that now, since everything has been fitted, I want to save this PDB. So what I do is file and use the save option here. And then you see the files of type, many types of file can be saved here. And you can see JPG, image, or anything. For example, but I want to save this PDB, of course, this whole PDB, which I have already fitted. So I'll keep this as PDB. And then I will say that, for example, let's keep it something name fitted model. Okay. I say fitted model. And then you have to, so you save model this. You see, this shows the save model. So because this is the second ID of the chain, and this is the PDB. So W1WA3, right? So you select this, and then you say, this is very important, because this is the final thing that you have to do. You have to check this box. You have to save this relative to which model. So we have to save related to this map, right? So this is the EMDP map, and the model, this has to be saved in relation to this, so that it remembers where we have fitted it. And then you kind of press save, and then it gets saved. The advantage is now it gets saved if you want to open again, it will open in this position. So if you want to directly open from here, the map and everything, it will come directly, fitted model will directly open like this. And you can directly use this fitted model to open it in Qt and do final refinements also, right? So this is how the fitting is done in map. Yeah, if anybody has any question, please free to ask, because I'll now be moving to some other section of AlphaFold. If somebody is not able to follow, or if somebody wants to just put up a question before I move to second section. Did everyone follow along what I was telling? Can somebody? Yeah. Okay. So this is how we do it. And is there any question? Okay, no question now. So I will just close all of them here, right? So you close all the model from here easily. And now somebody has asked, Samreen, if you want to save it as JPG, will it be the same? I mean, of course, JPG is an image file. It's not a PDB or relative model. So this is just for screenshot, for example. If I, for example, I'll show you, if I just open this fitted model, I mean, not fitted model, sorry, this is something else that I opened. If you open the 1197, for example, right? So this is the image, and you say file and you want to save it, right? And then you select JPEG. So this is just an image file of this. So this will not be saving a model or something. It's just an image file. And for example, if I bring it to my desktop, and you say that it's desktop, and you name it image, or whatever, I just don't care about the spelling here. So you say save. So it will bring probably this image somewhere. So it's not a procedure of saving the files actually. Yeah. So yeah. So let's.
I wanted to comment one more thing. Sorry to interrupt in between. So like we can also like kind of also like it's good to save the image, but also we can save session of what we are doing right now. Exactly. And that is actually a good idea. And that is actually also possible in PyMol and also in this. Yeah. And it's good idea to save the session, because then when we want to later continue our work, we can just start from there only. So we don't have to begin with the scratch.
And for that, as Sabah said, it's an option here in ChimeraX is save. And when you go right, you should. So it's a ChimeraX session. It's called CXS file. So once you save the session, it gets saved along. So you directly open from there. As Sabah said, you see directly the map and the model also fitted here. Whatever I was working on, I went to a T, or I want to work tomorrow, for example, then I just save the session. And once you open this session, unfortunately, this session, CXS file cannot be opened by Chimera. So this is little unfortunate because they have removed this compatibility. But I think this is something that you can do. And there are also other options that you can play around and you can save, right? For example, you can save TIFF image and other options, faster sequence, everything. So yeah, so let's close this. Now, this is the next part, and I now want to move on to AlphaFold using, right? So you might have heard about the AlphaFold, right? Which is an artificial intelligence now, and it predicts the structure directly from your primary sequence. So you give this the amino acid sequence, and it gives you the tertiary structure of a protein. And I have to say that I was looking into some of the models and that I'm working on with AlphaFold, and it I have to say, works pretty fantastic, and sometimes even better than what you can predict by crystallography. So there are some people who are working on this, and they say that sometimes it happens that you have a wrong crystallographic structure or a wrong cryo-EM structure, but rather a good predicted structures. So there are many people who are working on this, and they're trying to compare the structures and everything. But yeah, let's get going with AlphaFold, how this AlphaFold has been incorporated into ChimeraX. So this Chimera AlphaFold was not there in Chimera. And now, because AlphaFold just came recently, now people are working on this a lot, ChimeraX and everything. So what I'll do is try to open this PDF. I can send it along later, but for you guys. But for now, I will just show you simultaneously how to do the things, and then yeah, I mean, you can ask any question that you have. So I'm going to follow this PDF, and I'm going to open a EMDB map, which is a 3.4 angstrom structure of a mechanosensitive human ion channel. Okay. And the interesting thing about this molecule was that it's a dimer, and so you have to be careful about the caps lock. So you don't have to use caps lock here to open, right? So you open this, the code of this molecule is 30945. So you open this 30945 from EMDB, and you'll see it's fetching map now. So you guys can also try to open this in your computers if you have downloaded ChimeraX. It takes a little bit time because you can see it's receiving the megabytes. I don't know, it depends on also on the connection that you have with your MacBook and everything, but yeah, I think it's done. Okay, so now you see the map opens, right? 30945 30 and I open something else. Great. So it's 30495, I'm sorry. So it opens quickly because I was thinking why it took so much time for me to open. Anyways, I will close the previous one. So, or rather, I mean, to remove the confusion, you just open again. So you see this is the molecule, open 30495. I mean, if you are in the command line and if you press the arrow keys, you can also go to the history. So this is, you can use the arrow key. So 30495 from EMDB. So this is the map, okay? And I will try to play around with the, please unmute yourself if somebody is, when you're not speaking, because I can hear some background. So this is a mechanosensitive ion channel of a human, okay? And you can see this is a nice membrane. It's a two helix that are running together, intertwined. And you have this protein here, okay? So what I'm going to do is that I'm going to predict the structure of this, because there was, when people actually started working on this, there was no model for this. So of course, when you have no model for this, you have to determine it by AlphaFold. And so what people did is to use AlphaFold here. The authors use AlphaFold here in this. So you go to tools and you go to structure prediction here in the ChimeraX. And once you go to tools and then structure prediction, you have AlphaFold here. And once you click AlphaFold, it opens a new tab here. Now just look at carefully at the options that you have. One of them is fetch, the other one is search, and the other one is predict. So this is also in my, I have written this, this is also in my Word file, which I can send you along. So what happens is that there is an EBI, which is a European Bioinformatics Institute, which maintains this AlphaFold. And in the AlphaFold databases, if you click on fetch, it predicts the best. So you have to press the sequence here, and it will predict the best sequence of that, the primary sequence, and give you the structure here. Okay, this is one thing. Then there's an option called search. What will search do is to get you a list of sequence. So it will not find you the, for example, it will give you a list of 10 most similar sequences in the order. So it will give you the highest matching is number 1, number 2, number 3, and it will give you, for example, 10 models. But if you click on fetch, it will give you only the best model. So for example, if you have 10 models in the databases which are correlated, fetch will give you only one model, which is the best model. And of course, if you don't have any model still in the AlphaFold, then of course you cannot use fetch and search, and then you have to use predict. And once you use predict, then de novo it predicts, because now the AlphaFold knows that the sequence that you are giving, there is no molecule already in the databases. For example, I'll give you an example. So I'll later come to that also. But you can see in my screen, if I just enlarge it. So you can see there is around 21 organisms for which the AlphaFold database have the data. For example, Arabidopsis, swarm, number of predicted structures are already given here in this table. But for example, some of them, I will also give an example of this. For example, a chicken, right? You cannot see a chicken name. So there's no chicken structure deposited. So of course, for those you have to predict, but I'll come to that. This is a human. So, and you can see humans are there, right? So there are structures of humans deposited in AlphaFold, so organisms. So it's known. So what I'll do is to use the primary amino acid sequence of this and then try to predict it. So of course, this is the UniProt. So you click here. I mean, you can use directly because I have linked it to my Word file, so you can directly click. But if you want to, you just open UniProt, right? You use UniProt, you open the UniProt, and then in the UniProt, you type this code for molecule. This is, of course, there. So, and once you do that, for example, then you use this, go down here with this ID. So the UniProt ID for this is actually this Q9BJXJ8. So this is the UniProt ID. So if you search here, you will get this. Okay, so you can open the UniProt, and if you open UniProt and type this, you will come to the same page, which I am currently. Okay, so I'll show you UniProt, and I go to UniProt, and then I type this, right? So, I mean, I can just type it here because it's already open, but just to show you guys. So it's Q9BXJ8, and you search it. Okay, and so it opens directly this page because now it's already open, so it will not open. But if you type it here, it will open. So now you go to, I mean, there are a lot of details about these proteins. It functions. Their UniProt is known for that. It gives you what parts of the protein are transmembrane, what are the where the location. Some of them are cytoplasmic, but I don't have to worry about this, right? So what we'll go do is to go to this FASTA sequence here, right? And once you get the FASTA sequence, so this is the amino acid sequence of this protein. So you start with methionine and ends with aspartic acid, as you can see. So I'll just copy this sequence and say copy, and I'll close this Chrome, and I'll paste it here, which is the AlphaFold, right? And I put it there and I paste it. Okay, so now what I'll do is to paste this, and I want to select the best model for this because I know humans, this is a human protein and there is a human database in AlphaFold. So I just press fetch. And now once you click fetch, it shows you searching AlphaFold database for one sequence. So it starts to search now, and I see that's already found one of those. So you can see this is the structure that it has. Of course, this is a monomer. So now actually the disadvantage of AlphaFold is that AlphaFold can only predict monomer, and this protein is a dimer, right? So AlphaFold doesn't know that how this dimeric contacts will be. So this is, but of course now people are working on getting dimeric complexes and with the AlphaFold also. So now people are working on multimer predictions also, which is there and which you can read when I send you later. But generally speaking, it works only for monomer. So this is the one of the monomers. And the color is also useful here. So it's not that it's just random color, but this AlphaFold model, which has just opened, tells you how much confidence it is. So this, the blue part, the most blue part, tells you that this part was predicted very confident. So it's like 100% almost prediction. So it's correct, but this yellow and the other red part of the blue, for example, this tells that the confidence is pretty low, for example, 10 to 20%. Maybe because these proteins are very flexible, or they don't find any homology model for this when they search in the AlphaFold. So this color has an importance. So you have to remember this. And now what I want to do is to again fit this, right? And and see how it works. So as I told you, right, in the previous fitting also, how to fit it. So what I generally like to do is to change the opacity so that I kind of see the map, right, more clearly. But I have to say one thing, that generally what happens is that when you want to fit anything in this such a high resolution map, because you see it will be difficult to do that because the features are much more in better resolution and higher resolution, but then fitting is always easier in low resolution map. So this is what we generally do in molecular in structural biology, that when we want to fit something, we of course use a low pass filtered map. So you use a Gaussian low pass filter map and then fit it, and then of course you get a more reliable fitting because you can see the details here, but to fit a molecule like this into this will be a difficult task. So what I do is to generate a Gaussian curve, and for that I use this command, which is here, right? So it's a volume Gaussian function, and you generate a simulated map, Gaussian map, which is a low pass filtered map of this, so that you can fit this model into this map easily. This is the reason that I'm doing it. So I use volume, then you use Gaussian, right? So and it's written also here in my PDF. So I generate the standard deviation 1 and 2. So of course, in my case, it's showing the map 1, ID 1, but in my case, it's 1. Yeah, it's again 1. So I will use 1, and then standard deviation of 2. So it creates around 8 angstrom map. So did I do something wrong? Yeah, I think I typed the spelling wrong, right? It's G. Okay, volume. Sorry. GAU. Okay. Okay, so it generates a Gaussian map, right? You see a third, another map that has been opened here. And for ease of this, ease of you guys, I can do one thing. I can hide this for now. This EMDB, the low resolution, the high resolution map, which was more featured. So I generated from that map, the high resolution map, 8 angstrom something map, but you can see the path of the alpha helix become more clear when you have a low resolution map rather than a. So when you want to see the connectivity, always the lower resolution map is better than a higher resolution, but it's just, of course, for fitting and for that. But of course, you want, when you determine the structure, you want the highest resolution possible. So for example, 3 or 2 angstrom, the lesser the value, the better, right? So you can differentiate. So, but I'll use this, right? So to fit it here. So again, I use the same method which have been talked about, but again, as I told you, I like to make things a little opaque so that I can see more clearly. And then you can also play around with lights if you want, whichever fits. For me, this one looks okay to me. And then you press your control in your keyboard, and then you press the left and select any residue. So you see that one of the residues have been selected. You go similarly to right mouse. So rotate model is already selected. So I just use my right mouse now to rotate. And as I told you, you can simultaneously use shift to press the shift and then use the right mouse to move, right? So it's basically a combination of rotation and translation that you play around, and you bring this molecule towards it, and you try to align it, of course, by your eyes. Okay, I have to say that it looks okayish. And of course, finally fitting, you do, as I told you, go to the map and you press here fit. And you see now whatever was, once you bring it near, then it's easy for the ChimeraX to fit it. You cannot do the fit map button by placing the model here and you press fit. No, it will not. So you have to bring it to the region where it's pretty close, and then you press it. Can we use this software for material science research? Yeah, I mean, you can use actually more, not material, I have to say more medical research, I have to say, than material, because now people are using for generating some image. And I'll come to that actually. So there are options of medical images here, for example, generating orthoplanes, and even you can see brain CT, right, which is a CT scan of your brain and chest. These things are there, but I don't think currently material research is there. If you are more specific, you can ask me what material research you mean. I mean, which molecule you want to see. I don't think materials research is currently facilitated. It's more like biological research, and now biology is going towards medical image in ChimeraX rather than simple molecules and everything. So anyways, so now you see the fit in map function here, and it perfectly aligns, right? So what I do now one thing that you can see if you notice this fitting carefully, is that this aligns pretty well, right? I mean, almost. But I have to say this alpha helix doesn't align properly, right? So you see, this chain is running in a very haphazard way, right? So it goes like this, but it doesn't fit, right? So, and this is happening, you know why? Because this is the model that I've taken is from AlphaFold, and AlphaFold only predicts a monomer, but this protein is a dimer. So AlphaFold doesn't know how these two molecules, when they come together, so this monomer and this monomer of a molecule, when they come together and they interact, at what surface they kind of interact. And because of this, the AlphaFold cannot tell you, and that's the problem, and this is why it is happening, right? So how to resolve this problem? So one of the ways that what we do is to select these residues and then fit it independently of these. So for that, what I do, I mean, of course, you can try to remove this AlphaFold so that you just save the space here. And then you can hover the molecule. Okay, so you see this is, I mean, if you keep it here, you see that it's a proline 3. If you keep it here, you know it's a chain A and it's a methionine. So it's the first amino acid, right? Because protein starts with methionine. So you you can hover over this and you can see it's which molecule it is. And when you close over here, it's around lysine. Okay, so it's 99. So let's select around 100 residues. So you know that its chain is starting from, so this is the N terminal. So it's 1 to 100 residues, which are not fitted perfectly right now. So what you do is to use cell 1 to 100. And once you click this, you can see that this chain gets highlighted in yellow, right? So it gets selected, this part only till 100, not these which are already fitted. And once you select this, you want to fit it. What you do is to use the fit in map function. Now I'll just close it so that I bring it again. So now when you want to fit this, you cannot use this function which was here, right? Which is here, but rather you have to go to tools and then again fit in map, and now it opens, right? So what do you want to fit? So you want to fit only these selected atoms because this is already fitted. So you want to fit selected atoms, which are 1 to 100, and you fit in this map, which is a low pass filtered map, which I've already generated because I've closed the high resolution map already. Okay, so you see carefully what I'm going to tell you. If I say fit, you see the movement. So everybody is requested to see on my screen what's going to happen. And I press fit, and you see what happens, right? So it fits this residues perfectly now, but it misaligns this fitted, right? As you can see, right? So it was initially aligned, but the software tried to align this, and when it aligned this, it misaligns this, right? So this is not the way that you do this. Once you know that, okay, this was already fitted perfectly, and you want to select only this. So I'll just do undo. Thanks to this undo function, so I can go back and I can see that this is already fitted nice, and I want to, this is already selected, and I want to fit only this. So what you do is to go to options here, okay? And once you click options, you have move all molecules, because when it is fitting the map, it is moving the whole PDB basically, and you don't want to do that. You want to move only this 100 residues, and you want to fit them because this is already fitted, and you don't want to touch them. So you uncheck it. So you say that ChimeraX, that I don't want to move the molecules, and then you say fit, and it perfectly fits. And now you see it doesn't move the other atoms, right? Other atoms which is already fitted, and it fits perfectly. So this is how you do it, right? So by doing this. And now what I will show you is a final thing, just to appreciate the fact that how this AlphaFold works. So what I'll do is I'll select this, I'll remove this selection, which was 100 atoms, and I'll also select this, and I will remove this map from here. So I'll select this and say close. No, so it will close everything. So rather I will use this. Okay, so I'll use this, so the map goes away. And now you see the AlphaFold model, which is the monomer, which I already got it. You know how to do that. So let's open the PDB, which the authors have reported, okay, for this monomer. So of course, when you deposit any map and a model, you have to deposit map as well as model into the databases. So for this map, which I just showed you, this is the mechanosensitive ion channel. You want to do that again. what you do is to open again the things, the PDB that has been deposited. And for that, I think the code is we'll have 7CXR. So it's 7CXR. So when you click open 7CXR, it opens the PDB which the authors have deposited. And now what you see is that it aligns kind of perfect. I mean, one way of aligning it, if it doesn't align in your computers, is to use a command in Chimera, which is called matchmaker. And matchmaker kind of compares this two PDBs which are open. So I say matchmaker 1 to 2. So which means that I'm telling the ChimeraX to generate. So now it match makes, right? So it does it, and it also calculates the root mean square deviation between. So basically your alpha carbon, right, of these chains, it's how much different it is calculated by the RMSD, and the lower the RMSD value, the better it is. So it's 1.1 angstrom something for our case, so the RMSD. But what I mean, you can also do one thing, because this is a dimer, right? This is dimeric structures, as I showed the map, and that's why when you hide this AlphaFold model, you can see that the authors have deposited this dimer. But the structure that has been predicted by AlphaFold is monomer, and you want to compare it with the dimer. So why not delete this other chains, right? Which is so that it becomes more clear to us. I can hide it rather than deleting. So I just hide chain B, I'll say, and I'll say ribbon, because it's a ribbon. Sorry, you see. So now I have just deleted the B chain from the authors. And now I have only two things. I have an AlphaFold model, and I have a model which has been deposited by the authors. And what you see is that most of them actually aligns pretty nicely, except that I have to say that this alpha helix, this separates right from here. So there's a minor difference, but most of it actually are clearly similar. So this is something that you have to appreciate about AlphaFold. Yeah, and I think that's the end of this part of AlphaFold. And I can also show you, I'll just briefly tell you something for which, for example, this was the case in which human, right, which was known, that I told you that humans databases have been there in databases, but for some cases it is not. And then you use this predict button, which I was talking about. So if you close all the models, everything is clear till now. Anybody has any questions till now? I cannot see. Does anybody has question? Then you may ask. Or is everything okay? Everybody is following what I'm saying? Can somebody tell? Yeah, okay, perfect. So now I will open another molecule, which is, so I will open this 23883 from EMDB. So it's a electron microscopy database. So it opens the map, and you go to step 1 and you see the noise, and so it's actually a chicken transporter. I think it transports fatty acids, and this is the Fab. So it's an antibody which is bound here to this micelle bound membrane region of a transport proteins. So, and then I will now for this, I will open my sheet, which is the PDF, and the UniProt sequence is here for this. So the UniProt ID is F1NCD6, the sequence file for this protein. Similarly, I go to the FASTA sequence, and I copy the amino acid sequence of this. And where do we paste it? You know this, tools, volume, structure prediction, AlphaFold, and you paste your sequence here. And since you already know, I mean, you can try, but it will not search. I mean, if you press fetch, you see, you can try doing that. I mean, because it's an already deposited structure generally in the databases, it shows you the best one, but it's not a chicken one, of course, because this is something else that it showed, and I can tell you how. So this is not a chicken model that it opened. It chose the best model that was there in the database, but not from chicken, and that's the problem. So what I'll do is to neglect this, but rather use search, so you know what was this model all about. And when I press search, it starts searching. Right, so did it start? So okay, I lost the control. Wait a minute.
Sir,
Yeah.
sir, I just wanted to know how will we determine the which uniport sequence we have to select? Like here you took the transporter and you entered the fixed ID. How will we will know that this ID is we are going to look for?
Good. So what you do is you know the protein that you are working on, right? So generally structural biologists know that I'm working on this protein. So for example, I will just open some random. Let's talk about, what do you say? Okay, any heat shock protein. For example, I'm working on, suppose, a heat shock protein. Okay, heat shock protein 15. So I type heat shock protein 15. Okay, this is HSP 15, and you see this uniprot right here, uniprot. You can directly also type here the name, also it searches it. So you see the heat shock protein, right? And once you know this, you don't have to know the sequence, this prot ID. Once you, because you have to just find your protein there. You can remember later for later stages that if you want to retrieve the sequence, this is the uniprot ID of the protein. But of course, when you are working on a protein, you know the name of the protein. And once you know this protein, of course, you can scroll down and you can get this faster. And you can use this, copy, and paste it here and find, right? Easy?
Yeah. Yeah,
And
thank you, sir.
and the structures, of course, you know, because structures you have to determine by crystallography or cryo. So structures you will have, and the model you will get it by AlphaFold. If you don't have, of course, AlphaFold is used only in special cases. You don't need it every time. Okay. So
Thank you, sir.
yeah, welcome. So yeah, I mean, I can use this predict option now. So once you use the predict, because you know this as a hen, it opens this AlphaFold run software. So now you see that it tells me a warning that, okay, this ChimeraX, this notebook was not authored by Google. So it tells you that, okay, this is the script that you are going to run is not basically a part of ChimeraX. It's not a part of Google Colab, but it's a part of ChimeraX. So it gives you a warning. So that's fine. You say run anyway. And then it searches the sequence which you have started. And yeah, I mean, you'll see some things which are running now, and you can so this is not a paid version. You can see that the sequence length is 528, and now it's installing one by one on the collaboration. So it's everything is going on in the cloud of Google. So it's a Google collab which has been developed by with the company Google, and the AlphaFold database are there. And now it's doing on the cloud. It's actually doing some Python installing. For example, it's installing matplotlib, it's installing AlphaFold on the cloud, and then it will, from your sequence, it will generate the model of your protein, the sequence that you have given here. Of course, it's a very time-consuming process, so I'm not going to show you because it takes for, like for example, I think if I remember correctly, it takes around 700 amino acids. For 700 amino acid protein, it would take you around 2 to 3 hours, I have to say, just to run this. So it will run for 2-3 hours for a 700. And I have to say that if you go to around 800 amino acid sequence protein and then paste it and run it like this, most of the time, in my experience, it kind of fails in the sense that it runs GPU, runs out of error or something. So for that, people are using Google Colab Pro version. And of course, this is a paid version. You have to give around $10 per month, roughly, if I remember. And it gives you a timing of around 11 to 12 hours per day. And then you're not limited by time. And also, I think the amino acid sequence, you can search more rather than just 700. So yeah, I mean, now it will install and it will do everything. but finally it will look something like this. I will because it will take 2 hours, which I cannot do it in workshop, but I can show you how it will look. So this is how it will look. So this is the screen AlphaFold, which already was opened till here. So it will install this structure animation. It will start to search all the sequence database in the AlphaFold. It will take 29 showing, and then it will align. And then finally, it will give you a folder, create a folder in the normal Chimera, wherever your Chimera normal directory is. In my case, it's desktop. And it will give you some files like this. And in this, the best model.pdb, the first file, this is the model, AlphaFold determined model, which you can use. And this is the final result of the AlphaFold that you want to use for this kind of structures, because of course this is a chicken and you don't have a chicken database in the AlphaFold, and so you can predict rather than using the other two options of fetching and searching, right? So I have other things in the databases. For example, it can predict single protein, not complexes. You know, I told you this AlphaFold. Of course, there is a Multimer article 2021, which you guys can read how this paper predicts. And there are some problems, like I told you, runs out of GPU for long sequences, right? And I also told you, and it's also the fact that AlphaFold does not handle ligands and ions and solvents. So this is the problem. So if you have a ligand attached to your molecule, then it will not find it. And it requires expensive things, large databases. So this is something that I wanted to show you guys. Yeah. And I think that's the end of the AlphaFold part of my talk. And yeah, it will run for long, 2 hours. So I'm not going to let it run. I just close it. It was just for showing how to do that. Yeah, I think that's it. So we'll take a 5-minute break, I think, and then we will come back, hopefully. If anybody has a meantime any question, then please feel free to ask now, or you can also type in the chat box. Yeah. So we'll meet another after 5 minutes, hopefully 5-10 minutes. Okay, so we'll come back after break. So we discussed 2 things, right? So if you remember, fitting the densities and everything, and then we discussed about AlphaFold, how to use AlphaFold to determine the structures. Next part, I will quickly show you 2 things, and then I will work on that. So let me again share my screen, because I think it's not visible right now. So now the third part of my the session is to show the residue fit. And I mean, you can follow this in my PowerPoint slides also. I can share it later. But how do you do it, right? So this is the question: how do you show the residue fit in your crime density? In this is actually done in mostly in Qt. It's a different software altogether, I have to say. It doesn't work. I'm not going to show you Qt or anything right now, because it will become a too hectic thing. But definitely I can show you a short method to do that. So you open a map and a model. So I will open first PDB. So you open 5MV5, and once you open it, so this is the PDB of that molecule that has been opened. Okay, and you can see there are some ligands also. For example, this here. So if you click here, it shows you it's a ligand here, and another one, I think here somewhere. No, it's not. So this is unbroken chain. So you have one of those. So I opened this 5MV5, and I opened the corresponding map for this. So it's a 3574 from EMTP. And you see this is the map, and I go to the level 1. And of course, now the model is inside this map, so it's not visible for you. I mean, I will discard this AlphaFold so that it's not creating confusion. And I mean, you can do one thing. You can, for example, move it to the transparent side so that you know where your model is. Now you can see the model clearly, right? It's fitted somewhere. And there are many copies of the same thing. It's all around. So there's a symmetry again. but I don't want to touch into that. But what I wanted to show you is how to show the residue fit, because now if you want to clearly go into each of the residues and see whether each of the residues is, because this gives a very holistic picture, right? This gives a very overall picture that your molecule looks fitted. But if you want to do it in a way that you want to go residues by residues and taking screenshots and probably making movies of this fit, then you can use this tool. So that's called res fit in the ChimeraX. So I will use this res fit. If you type res fit and you say that I want to do it in the 1, which is your because ID is 1 of the molecule. This is the PDB of ID, and you say of chain, which chain, you say only chain A, because this is the chain A that has been opened, and you want it to fit it with map, which is already open and which is localized in the second ID of the ChimeraX. And once you type this and press enter, you see this kind of thing. And now when you bring it to the circle, you can move the threshold of the map, right? So because you see the glutamic acid here, right? And now when you try to play around with the threshold, you see a better density fit, because now you see that it's not, yeah, I would say now it looks more or less fitted. Now you can more or less, you want to determine the basically a suitable threshold through which you can see other molecules also. So that's why I'm playing around with this threshold. So I know at which threshold the density more or less looks like fitted. So this looks like fitted. And now you see that when I press res fit, it showed you a residue fit. It opened this tab here where it already goes to residue. If you press up button, it goes to the next residue, serine 4, arginine, aspartic. You see that, okay, this chain is fitted perfectly. You go to 7, you say threonine, okay. So from this cage, it was so dense a structure, but right now you can see at an individual level of your amino acid how it is fitted, right? So this is one way of doing this. The another way of using this is using this play button. And when you click this play button, it gives you all of the residues one by one. So it shows you the third, and then it shows you the fourth, and it simultaneously goes around one of those residues simultaneously, and it shows you how the fitting has been in all of the individual residues, threonine and then valine, for example, at 9. So it will complete the whole chain from the second amino acid to the 253rd amino acid. But I'm not going to show you because the interface is, anyways, the main thing is that I wanted to tell you was the function. And if you press this button now, which is the here red, and if you stop it, and then you press pause, sorry, that isn't it. And so press stop. Once you press again stop this second time, actually it generates a movie here on my desktop where it is here. And if I click this, second, my Mac also is taking time to open it, although I have 16 GB of RAM. You see. Anyways, so now you see this is the MP4 that has been generated on my desktop because I generated a movie out of this. And you see the fitting of my residues. And of course, when you want to move it to the center, that's better. So that you don't see this density on the side of the screen, but rather on the center of the movies. That you have to align when you are making movies. But this is how it's done. So you can see that I can basically see all the residue fit in my density. So this is one part. And I think most of the thing is clear here, right? Or if there is any question, please feel free to ask. No? Okay, so then I move to the last part, which is coloring and making models. So what I do is to show you guys how to generate a movie, do a coloring, and then of course record a movie for that. So I will open a structure of a very multimeric protein. So it's like easy to do that. And when I do that, I say it's open 23526 from EMDB. And this is the structure. And now you see the map. It's around, I think, 4 angstrom or something, whatever. So you go to step 1. And you can play around with the threshold. Again, you see the noise coming, so you don't want to go there. something like this, right? This looks like an okay chain. And now you have to, because there's no chain here, because this is a map. So, I mean, you want to open the PDB for this, which is already deposited, which is the model for this. So it's called 7LUP. And once you open this, it opens the model. And I generally prefer not to have this atom view. So I just hide it and I show the cartoonish view. And of course, there's everything is just hidden. So when you see the opaque, you see the fitting, right? So you see the fitting now, that the model is there and the map is there in the fitting. So you can use the other views like soft lightning to generate more three-dimensional view and appreciating the fact. So this is the map in the model. Now, what I want to do is to map, you want to color near the atoms, basically. Okay, so you have a different chain right here. And there are, I think, 8 or something chains. And you want to color these things according to the atoms. Wherever there is an atom, it will try to color it. And for that, you have an option called map, and then you have color near atoms. And you press this. When you press this, because the PDB was there already, so all of the chains get colored differently, as you can see, right? All of the chains, it get colored differently. It's a surface, so, but you can see this. So this is a way to actually color the proteins according to the chain in a different way. So all of the map gets colored here, and all of them gets colored into a different way. So this is the one way of doing this thing, which is pretty easy. Another way of doing the same thing is to use the other options available in the tools. So for that, I will bring this back. So I want to make the color uniform again. It was, so let's make it any color. Let's make it blue twice. So now it's again in a uniform color, right? So it's not a I will make this non-opaque now, so that, because anyways you saw that it's the model is fitting perfectly. So this is all uniform again, right? And now you use an option called color zone. So you go to tools, and then when you go to tools, you go to volume data, and then you use color zone. And when you press color zone, you see this thing opening, right? And this is the tab, and it shows you which surface to color. So it's a color surface, and near what. So it tells you that. Okay, so I have selected color surface, this map near this PDB, of course, and this is the coloring radius. So when you press color here, it colors the atom. So this is another way of doing it. And of course, this is a threshold. This is a normal threshold of 7.8. So it determines around whose atoms, around all of the atoms, 7.8 angstrom it covers, and then it tries to put the color around that. But of course, you can also select a single chain and you can color them rather than coloring them differently, right? So if you want to do that, you have to do select, right? Select, for example, and then you press chain A. And now you see, if I hide this, there is some chain which has got selected here. You can see, right? This is chain A which got selected here, and I want to color them only. So I just want to uncolor it, make it normal, and you see that the chain A has been selected. So I selected the chain A, and I want to color it only here. So I will go to the color zone, same option. If I want to close it, again, you can open it by tools, right? And then volume data and color zone. Now I'll say EMDB map in the selected atoms, because I've selected only the chain A. I don't want to color the whole map, but I want to color the surface only near the selected atoms. And when I do that, you see that the map gets colored, but of course it overspills also. What I mean is that apart from selecting the chain, you can see some extra chains getting colored. And of course you can play around with your densities here, coloring radius to get rid of that, right? So it gets, the only chain A gets, for example, selected right now. So you see now the chain A is getting colored. So this is the, I would say, optimum coloring radius to get this chain only. So you can color other chains also. There's a command line for doing these things also, but again, whichever is does the same function actually, this color zone function. So you can do this color zone, right? So let's make it uniform again, right? So that I know which is this. Now I select 3 chains, color zone, right? 1, so the first atom, and map near which A, B, and C. So the A, B, and C chain, and you say distance. So this is the distance radius here. So the normal was 7.98. I do it half, so 3.98 something. Okay, so I say that, okay, color zone, the map 1, which is this map, and you have to color it near the chain A, B, and C. So the chain A is already selected. Chain B and C will be somewhere like there or there. I don't know where it is. But I tell the camera that, okay, you select all of those 3 chains and color all of the atoms near the chain AB with a radius of around 3.9 angstroms. So I say it, and you say it colors the chain A, B, and C. So 3 of them get selected, and the A, B, C gets highlighted. The other doesn't get highlighted. And now, of course, if you want to use make movies from that, you can go to home. There's an option called spin movie, and when you click that, it will take time. I don't know, on your computer it will be a little bit troublesome task, I have to say, because it's memory intensive, I have to say. But you see, now when I say the spin movie, it's the interface says that it's capturing frames. So it will capture 180 frames and then it will generate a movie. Of course, you can change the speed and everything of this movies also. There is like a function in the help function where you can do all of these things, but just to show you how this works. And it make movies out of it. And then you can, people are doing it nowadays on Twitter, that they're determining the structures and then they're generating movies through ChimeraX. And then they are putting it on Twitter to show that, like our lab has, for example, determined the structure, and it gives you a very decent look because it generates a movie and it gives you a 3-dimensional structure and fitting everything. And then you can show with that nice coloring. So you see this movie here on my desktop, which has been generated. If I click here, it's taking time to open. It will open soon, I think. Yeah, now. Yeah. So now you see it opens my media player, ChimeraX, and if you place the play button, it shows you the movie, right? So of the atom fitted. I mean, of course, this is non-realistic because I have generated a surface view. People generally display the transparent view so that you see the nice fitting and of the chains, because when you have high resolution, this is what the structural biologists want to show, that you look at the phenyl chains, it's fitted perfectly, right, at high resolution. And that is why you do model building, that you know that, okay, at this resolution, at like 1.5 or 2 angstrom, you can basically trace the complete backbone, and you can even see the holes in the phenylalanine, for example, in the ring of the phenylalanine. So you know the resolution and everything. So this is how the people are using the movie function. And I think there are a lot of things to explore for you guys. I have to say that I cannot cover a lot of them, so I have to end the session workshop now. But there are many other things. For example, you can even color it by chain, for example, all of the molecules by rainbow, by electrostatic interactions for hydrogen bonds. Even you can hide hydrogen bonds. You can show the sequence through the PDB. And there are a lot of things, I have to say. It's just a matter of playing around with the things. And yeah, I mean, ChimeraX is I have to say, it's a recent development, and people are moving from Chimera to ChimeraX. And there are many functions now. There is also a function of actually virtual reality coming. So you can basically use your mouse, computer applications, as well as there are some mobile applications coming, which can actually help you to see the molecules in your mobile or in your screen in a 3D structure. So basically you will put the virtual reality, put your the camera on your eyes, and then you can see the molecules in 3-dimensional. So these are things are coming in the ChimeraX. And I think in the next releases of ChimeraX, the software developers will also reveal it. So this was the electrostatic, which I clicked. You can see the chain A because it was already selected. So it showed you the crevice, right? So the blue being hydrophobic and this hydrophilic. So different kind of things that you can do with these softwares. When you have nucleotide, for example, if somebody is working with a DNA or something, then you have rather than amino acid, you have adenine, guanine, thymine, cytosine, right? For example, if you have urine, then of course RNA, then you have uridine instead of that. So you use this kind of views. Then you have graphics, which I've already showed you. map functions. Medical images mostly we are not using because these are for medical people. And yeah, right mode functions, which I've already discussed in a brief to you guys. So yeah, I think that's all from my side. If anybody has a question, please feel free to ask now. So we'll take up questions in next 2 minutes if somebody has any question, and then we will wind up. No, I don't think they have any question, right? So if not, I mean, if somebody has a question later at stage, you can always email us at ssgsa. What's the email address? Contact, right? Sabab, I can tell you better, or you can email it.
Sorry, I will just write in the chat our
Yeah.
email address. Like
Yeah.
it should be. I will just write in the chat.
Yeah, so you guys can, if you want to ask any questions at later stage, you can always come back to us. But yeah, if you have any question that is coming into your mind right now, you're free to ask. And if not, then I give the stage to Sabaudin Bhai for his closing
Can we
remarks.
get
And
the recording
yeah.
of this session after this? Sir, can
Yes,
we get
of
the
course.
recording of this session?
Yes, of course,
Thank
that will be available. Yeah.
you. Okay. Thank you, sir.